The primary germ cell, spermatogonia, are transformed into mature sperm or spermatozoa, while oogenesis is the sequence of events by which the primary germ cell of the female gonia are transformed into mature oocytes, the one she shared at the end of the month. Are you? I hope you are following. So let's take. So let's. Yeah, Jesus. What's the question? Is it Jesus, right? Jesus. Who? What is the question? What is the problem? I'm seeing your hand up. I continue.
Yes.
Okay. Okay. So now you can explain what is gametogenesis. From gametogenesis, we have explained what really happened to the germ cells. Now, the formation of the sperm or the male germ cell and the female germ cell is known as spermatogenesis for the male and organogenesis in the female. So let's look at spermatogenesis. The aim is the formation of the sperms with haploid number of chromosomes, site in the seminiferous tubules of the testes. Don't forget, duration about 2 months. It takes 2 months for spermatogenesis to occur. Occurrence starts at puberty and continue through life. Through life, it keep on occurring till when you are 150 years. Silvio, you understand that?
No.
So these are the process. Can you see the next slide?
Yes.
Yes.
Okay, fine. So these are the simple diagram, all these things grammar I'm talking about. You can use this simple diagram to illustrate that this is the spermatogenesis. It undergo what we call the first primary division. After the first primary division, it undergo meiosis. The
Your question.
meiosis now, yes.
May you zoom that so that we can see the words. We're not seeing the words.
It okay. If I zoom it, maybe you can. The slide will not even move. Maybe.
We are not seeing the words. We're seeing only the diagrams
The heading.
only.
I'm
Yes.
trying to zoom it now.
Okay.
Hmm. I'm trying to zoom it. But it seems that the zooming is not show slideshow. No. Uh, layout slides. No. Okay. Let me go to show slides. So let me share. Oh, slideshow. Hide. Setup. Some. No. I'm trying to see if I can zoom it. Hide. Setup. Mm, It's not. I don't know what is happening. It's not coming, right? Can you see it there?
There is still no change now.
Can you see it?
We are seeing the like how we were seeing before.
Yeah, that is a problem now. Set up slideshow, Customize slides. 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 I don't know what to do, but maybe I was well. If you, I can explain, then maybe I can share the slide to you people. Oops. Yeah, what do you think, Arafat?
Yes, lecturer.
It's not this slide is not able to. Okay, maybe let me see.
So
Can you see it now better?
not not yet, sir.
Okay, let me explain. Maybe is it a bit better?
Yes, we
Lecture, we are seeing it. We are seeing the words.
explain it.
May continue.
Okay, only the word. Okay, maybe yeah. When I share, you can use. It's not too much slide, so that I finish before time. Here we talk about like explain what we call spermatogenesis, the formation of sperm from the male germ cell to a single mature sperm. Do you understand? So anytime you ejaculate like have a tea, one have a tablespoon, which is a teaspoonful. That's the quantity of ejaculant when if you are normal, and that quantity of ejaculant is 2.5 million or so ejaculant. If you quantify all, it's like 2.5 or to 5 million. When if you are healthy, you ejaculate like 5 million at a time. So that 5 million is like a teaspoon. You know teaspoon? Do you know teaspoon?
Yes, we know it.
Is the class not there?
Lecturer, we are still there.
We are there.
Yeah. Now nobody is talking like you. You never ejaculated before.
We did.
Yeah. This is a practical something. It's not. Is it a bad thing? It's not like this is a practical class, and you are all medical doctor. You are mature.
Yes, we
So
are.
I'm not sure there's anybody in that class, a male child that have never ejaculated. We started ejaculating from the dream. You understand? Then you never know how to talk to you never know how to talk to Makobga, but you have already ejaculated.
Yes, we had it, lecture.
Ejaculation doesn't mean ejaculation doesn't mean that you must talk to a Makobga or a woman. No, we have what we call the wet dream. The wet dream, in the wet dream, it happens where sometimes you don't know how to talk to Makobga. But when you have a wet dream, you learn all the skill to talk to Makobga in the wet dream. Do you know that?
Yes, lecturer.
Yes.
Yeah. Anybody that have a wet dream, when you wake up in the morning after the wet dream, even though you want to go and meet the president wife and tell her that you know what to talk to your wife, whoever, when you have a wet dream, you are bold to meet any woman, big, small, whatever. You can tell her everything. You don't even you have the spirit. The I don't know how I can explain, but I know you can understand with me for the male. Yeah, yeah, you don't you can you after you finish talking to anybody, you can say, "Oh my God, am I the one that went to talk?" But the person doesn't know that all this experience they come from the wet dream. You wake up in the morning, you talk to her in the dream. So what I'm trying to say is the fact that one ejaculant, the wet dream, that wet dream, the quantity under normal condition is full the teaspoon. So that teaspoon is total is 5 million. If you take it to the lab, you when you do just I don't know people you don't do it here, but we can you can take the sperm, you analyze it one after the other in chemistry, in clinical chemistry lab. But I here I don't see they don't do it, but we teach our students either they bring their own sperm or they remove the one from a laboratory animal and then they analyze it. You can see them moving very well in a separate way. They are running. They will keep running till they die in the you know what they call petri dish. In the there's a dish in the chemistry lab. You see them in the test tube. They keep moving and moving and moving. When they are tired, they can be weak and then they can die. You can see it there. So, but here I don't know. Maybe everybody is ashamed or they don't do it for the student to see. So, but I don't know why it is not done that way. So that is what I'm trying to explain. The formation till you get that ejaculate, then you remove 5 million or 4.5 or 3 million or 3.5 million. You put it in a test tube. So now you have one spermatozoa. That spermatozoa, what happened for you to get that one that is swimming is known as spermatogenesis. Is that understood?
Extra question.
Before the question, you see for this slide to form after puberty, it takes 2 months. So after every day, a process of spermatogenesis is complete. Since you get to puberty and now, every day you produce a spermatozoa. Either you use it or you don't use it, they are there. Is that understood?
Yes.
Yes. What is the question?
So I'm asking, how can we take that sample by ourselves?
How can you take the sample? By yeah, you. It's a good question. Whenever they want to do the experiment, we give you a test, just like when you go for to the hospital, they want to do exam. They can give you container for the exam, for the urine, kunyara, or the how can I say, and for the poop, for the pee, for the saliva, for the cough, even for the sperm, they give a container. Okay, of course you can stimulate yourself if you can stimulate yourself, but for you can stimulate yourself, or we can even get it from the rat test seminiferous tubules. You understand? But as a doctor, what do? You advise the couple together during the sex. Sometimes the male will come to you as a doctor that my wife is not getting pregnant. The woman will say that maybe they can blame the man or they blame the woman that she's not giving birth to a child. So what do? You examine the egg. Well, we'll get to that point. But for the male, you ask him to bring the sperm to the lab. But because you have not done the practical, this practical as I'm talking, you will not know the sperm that is alive and the one that is dead. Some men, as they are moving around, they have a dead sperm. You know the reason why? Because they put their phone. Hello, are you there? Somebody is talking. Who is
Yes,
that?
you are.
Are you listening?
Yes, you are following.
Yes.
Okay, you know that now you put your phone in your side pocket. There's a lot of microwave going on there. A lot of men very soon they will stop producing baby because the phone microwave is killing the sperm for sure. But people don't know, but that is a recent research. So, but if you have done a practical, in the practical in clinical chemistry lab or whatever, you would know the difference between a living spermatozoa and non-living spermatozoa. So many men in the nearest future will be having problem of infertility because the microwave for the phone is destroying the spermatites. So the process of is blocking the process of spermatogenesis. This process that take 2 months to form is blocking that processes. So that is one thing you have to understand. So in your question, it's easy to get the sperm. It's not a difficult. Even we in obs and gynecology, radio, urology, they teach you how they can. We can always teach you how to stimulate your penis to bring out the sperm for analysis. But people here, I don't think people are open. People are so shy. But during our time undergraduate, we do it. You understand? We do it. You stimulate the testing. You do it, and then we produce the sperm, and the student we analyze it in the lab. He say, "Come on, but I'm not sure people here do it. They prefer to do something secret. That is one thing I discover, and sometimes it's not helping your learning here. So that is one challenge I have observed. So, but that is how I don't know. That is the reality. But I don't know whether it is ethical or I don't know whether it is shyness. I don't know. But you would be the doctor to advise. You would be the doctor tomorrow to solve the problem. You understand? And I am only giving you the basic knowledge, what is possible. And if you follow it and do it for sure, you can counsel, you can guide, you can be a good medical counselor. Is that understood?
Yeah.
Yes.
Yeah. So to get the sperm is not a problem. All of us know as the male, we know how to get it. So the reality is that they will give you a container where you put the sperm with what we call it the physiological solution. It's just that I don't have time or I'm not too exposed to all those things. And maybe even though I am, some of you may not be. We don't have animals. If we have animals, I can do it from the animals just for you to see what we call sperm cell motility. Sperm cell motility, sperm cell viability, and sperm cell morphology. These are simple laboratory Yeah, maybe this the network is not too good, but we will manage. So we talk about spermatogenesis. Are you there? Are you following?
Yes, I follow you.
Yes.
All right. So you make it interactive. If you have a question, you make it interactive so that we explain. It's a very simple course. Gametogenesis is an interesting and a simple way. And if you understand it, you can counsel a lot of people and you can help a lot of family. And don't make sure you get the point. Clear, say the simple basic science. What happens? What used to happen? But because people are too shy, they are not open up. They can have a problem and even die with the problem. They think that there is no solution. They think the society is like it's a bad thing to talk about. It is not a bad thing. It's something. It's simple, but You are the doctor to solve the problem. So that's why I'm trying to be so open to I explain vividly and clear so that you understand the basics. When you understand the basic, then that is over. On gametogenesis, spermatogenesis, and the oogenesis. That is all, and that is all about the gametogenesis you have been looking at that way. The other thing can be practical. The other thing also can you follow it from the theory as you have it. So in this stage, in this slide, proliferation, each spermatogonium divide into two daughter spermatogonia, which is the with diploid number of chromosome 42 and the sex chromosome XY. The growth, then in terms of proliferation, then the growth is where spermatogonium enlarges for enlarges to form what we call the primary spermatocyte. And maturity, which is the maturity undergo reshaping and then reduce in number or increase in number, which is meiosis. So we have first and second meiosis, first division and second division meiosis, where we have in the second meiosis, the process of mitotic without a normal interface. That is to say, without DNA replications and things like that. So the next slide. So this where these are simple diagram to show you spermiogenesis. Spermatogenesis, there are two different things. Spermiogenesis is the competency the sperm is going to have to enable it have the energy, the strength to behave like the what we call the streamline or the point. Is it the pointer, right? Remember you mentioned the pointer.
Yes, that's the point.
Yes.
Are we together?
Yes,
Yes.
Rickson.
We're still with you, sir.
Hello, can you hear me now?
Yes, we
Yes.
are hearing you.
Okay. I said that there's a difference between spermatogenesis and spermiogenesis. The spermiogenesis, you understand? Remember we talked about the streamline, the stream shape of the sperm. So spermiogenesis is the forming apoptotic, forming the is the process that lead to the formation of a single spermatozoa by reduction in shape, building up the mitochondria that have the energy, align the mitochondria. Then when you ejaculate it, it moves with the speed it's required. The energy are built up by the mitochondria for it to move. So that is what is called spermiogenesis. So here is I said the seminiferous tubules. So this is what you can see the next slide, please. Can you see this slide?
Yeah.
Yes.
Yes, we are sharing it.
Yes.
Okay, fine. So here is the seminiferous tubule. So when we enlarge it, you can see that the seminiferous tubule contain at the base main membrane the spermatogonia. You can see the primary spermatocyte. You can see the secondary spermatocyte. You can see the spermatite, and finally capacitant. On the from the spermatite, it undergo what we call spermiogenesis to form the spermatozoa. The spermatozoa have the head, the neck, and the tail. So the head, you can see the acrosomal cells or the acrosome which carry the chromatin material. We see the head, we see the nucleus, and then the acrosome has a lot of enzyme that can digest the female ovum anytime, anywhere. And then after that, it will now secrete another secretion to protect, to prevent another sperm to fertilize it. That's the reason why the acrosome is very, very important. After fertilizing, it will stimulate the ovum to protect itself. No matter how many sexual intercourse you have, it will never allow it to touch the ovum. It will protect it against another cell. If not, you are going to have what we call teratology. Is that understood?
No.
What I'm trying to explain is that when one cell, you see the one spermatozoa, okay, one spermatozoa with energy capacitance, a, a lot of what can I say? A lot of energy due to the presence of mitochondria. After it has moved to the ovum and fertilize it, it will secrete protective enzymes. It will not allow any sperm to enter the ovum again. For example, the first and the strongest spermatozoa that touches the ovum is the one that fertilizes it and protects it. Is that understood?
Yes.
Now, are you seeing the slides?
Oh,
Yes. Slide 17.
Slide 17. Good. So if you
Yeah.
look at it, this is a complete spermatozoa from from spermatogenesis and spermatogenesis. This is the final product. Is that understood? Even in the laboratory, when we do experiment with other animal sperm, this is what we are expected to see. You can watch it here. You don't study very hard. Everything is easy. During our time, you remove this one either from the rat or the rabbit or from the dog or from your colleague. Then you label it. You can see it on the microscope. You label it and clear. Then you can see the head. This is a chromosome that contains the digestive enzyme that will digest the membrane of the ovum. After digestion, it will enter, and when it enters, it will secrete, or it will even digest the other part of the sperm out of it. Is that understood?
Yes.
It's just that there's no way
Yes.
we can do there's no way we can do experiment here because after fertilization, you understand? After one spermatozoon fertilize an egg, there's what we call vaginal plug. Are you following?
Yes.
Do you know what is vaginal plug?
No.
Okay. Vaginal plug is when one spermatozoa fertilize an egg. Okay. Then it will kill other sperm around the sperm around it will kill other sperm. It will secrete an enzyme that will kill other sperm. Then if the woman can be see whitish secretion every morning. I don't know. Are you following?
Yes, lecturer.
Yeah, after the first, if there is fertilization, the following day or the following morning, the woman will see a lot of white secretion. That is the sperm the first one have killed the other one and push it out to the vagina outing. In the morning, some maybe the woman may not want to show you the man, but she will experience it, and she may want to she will say maybe she will look at it, maybe okay, yes, I make sense. Maybe this is the man's spermatozoa, but that is what we call vagina plug. In dog and in lower animal, when you see the vagina plug, you know that there is pregnancy. You don't need to do pregnancy tests. I don't know
Teacher,
whether you are
question.
following. Yes.
Is it vagina plug or vagina broke?
Plug, vagina plug. You know plug?
Okay, yes.
Plug, vagina plug. We call it vagina plug. After the first spermatozoa fertilizes an egg, it secretes a lot of all the. Can you imagine you put in you ejaculate 5 million or 4 million sperm, okay, and all of them they struggle for only one ovum, only one. They struggle for one. Is the one that have the one that have done a lot of gym, have energy, is the one that will fertilize the egg among among the spermatozoa, among this slide you are seeing. You know you are seeing this slide, the sperm, right?
Yes.
This sperm you are seeing, this is one. Then I said that you are when you ejaculate one during woman heat period, you release 4.5 million of this one. Is that understood?
Yes.
So what I'm trying to say now, the acrosomal there's what they call acrosomal reaction. The acrosomal reaction will digest the female ovum and enter. Only one will enter. When it enter, it will cut the neck. It will cut the neck of the spermatin. It will cut it. Only the head will enter. When it digests, it will enter. Then the nuclear material will just enter inside. Then the reaction will kill all the remaining. Then it will kill other the remaining 4 point. It will kill you release assume you release 4.5 million. One will fertilize, then it will now react a chromosomal reaction will take place. It will now kill the remaining 4.49 the spermatozoa. It will kill it. So when it kill it, they will all come out of the vagina and block the vagina in the morning, or in the morning, it is after 12 hour or 24 hour. In the morning, the lady can wake up and see that there is white secretion in her vagina. Is that when you see that secretion, there is no need to go for medical exam as a doctor, as a gynecologist. You when you check that, there's no need to go for exam, because we are human beings. We like technology. We can go for exam, but if it is a dog or laboratory animal or rabbit, you carry the rabbit anus and you see the vagina plug, you keep it. You know that that is the day one of pregnancy. I don't know whether you are following.
Yes,
Yes,
we
sir.
are.
Lecture, not,
Do you,
not, we
you
didn't get it
are you
well.
pardon? Are you following?
Yes, we are.
Following, but not getting well.
Is my voice cracking, or is it you are not getting? Is my voice cracking? My voice
No,
is
it's
not
not about the
is not.
voice.
No, it is not.
Well,
Do
that,
you do you when you look at the slide very well? I am explaining what I explained. I have talked about gametogenesis. I've talked about the male and female germ cells. Then I took only the male germ cell. In the male germ cell, what the key thing you're supposed to know that is spermatogenesis. And the next thing you're supposed to understand after spermatogenesis is spermiogenesis. After spermiogenesis, this is what you have. This is the final stage of spermiogenesis. This is the final stage of spermatogenesis. Spermatogenesis in the male, gametogenesis, spermatogenesis, and spermiogenesis. Those are the key things components. You have to understand the different processes that occur at that point. Do you follow to that? Are you following to that point?
Yes,
Yes, we're
lecturer.
following.
Now you now produce one sperm, which is the most important this is a typical male germ cell. This thing you are seeing here, when you release it, there could be many. One of these could be all the X, the Y, they are very strong like this. The X could be like this, but they could be weak. And we only know them because the Y stay 48 hours. Why the Y stay for 48 hours? Why the X stay for 96 hours? Is that clear to that point?
Yes, that's very clear.
Then now, when I ask you to label a simple spermatozoa, this is what I expect you to label. Is it not true? When I ask you to draw and label, is it not what you are going to label for me?
Yes, it is. It's
So what you are seeing here is known as the dreamline structure of the spermatozoa. Is that clear now?
Yes.
Yes.
Now, what make it to move faster is we call it the end plate tail. The end plate tail make it to move the end piece the end piece of the sperm and the principal piece of the stem. They are the thing that give it the energy to move because they are all microtubules. They are microtubules. They are like they have to say they are microtubule protein, like the protein of your hair. Is that understood? Do you follow? I want to finish this slide now
Yes, lecturer.
because we don't have time. Do you follow?
Yes.
So,
The energy inside this sperm is gotten from the mitochondria sheet. Can you see the mitochondria?
yeah.
Yes.
Now, when you see the mitochondria, now you see the neck. The neck, that is the end, the neck, at that neck, that it will give it the energy. All the energy this sperm needs are gotten from the middle piece, which contain the mitochondria. Do you follow?
Yes. Yes.
Yeah.
Then the moment the head of the sperm, the spine of the sperm touches you see there will be acrosome. There will be acrosomal reaction. The acrosomal reaction, you can see the acrosome. That is called acrosomal reaction. These are protein, digestive protein that will digest the female ovum. That is why you see the acrosoma. It's
ariko
the
nanjye
first thing that will
ndiruka abantu
digest.
ushaka kubimenya fresh niba nangwa nzigarukira mu Rwanda mani bikoreye mu Rwanda mani
Is that understood?
urumva nanjye ubu rero
Somebody is talking from the
nibyo
background.
nibereyemo tu
yes. I have muted.
Okay. So can you see the next slide?
Yes.
So I've talked much on that spermatogenesis. So the next slide right now is the seminiferal fluid. The seminal fluid. The seminal fluid has a secretion from the seminal vesicle, prostate gland, and the bulbar rectal urethral glands. So the volume is of this seminal fluid is 3 to
zimwe na
5
zimwe
mil.
imirongo imwe n'imwe y'abahanzi igaruka kuri uyu muryango wa Illuminati igenda igaragaza ibimenyetso byinshi bitandukanye ubona bisa naho bidasanzwe.
So the sperm count I've told you is about 100 million per mil. And remember, the teaspoon is almost 2.5 ml. The tablespoon is 5 ml. The teaspoon is 2.5 ml, meaning that the ejaculant is almost 3 million ejaculants at a time. Just as I said, that 3 to 5 million average. Motility about 3 millimeter per minute. At least 70% of sperm should be motile. Abnormal form should not exceed 10%. So that's why, because we have not been able to do the experiment in the lab. If we did it, you would have seen all this. You would have seen all this thing clear. But unfortunately, I'm not sure it's possible. It's not my fault. Me, I'm always there to help. But if the facility is not available, I can't kill myself. So that is all about the male germ cells. So let's look at the female in a few minutes. Oogenesis aim at the formation of the secondary oocyte with haploid number of chromosome site in the cortex of the ovary. Occurrence start during fetal life, become completed at puberty, continue till menopause. So here is the diagram illustrating what I've just said. We see here it will undergo what we call the mitotic would have already been formed. So this is the first meiosis, and it will undergo first and second till the final stage is one ovum, and you have the second polar body, which is not always very viable. So here we say that during fetal life, under proliferation, we have each ogonium divided by mitosis division into two daughters ogonia. Growth ogonium enlarges to form primary oocytes. And then during each ovarian cycle after puberty, then it moves from first meiotic division completed, second meiotic division begins, second meiotic division is completed, and then there's no primary oocyte after that. So we talked about ovulation. So the ovulation is the shedding of the egg from the surface of the ovary into the fimbriae and into the fallopian tube for the sperm to fertilize. So that is what is called ovulation. Ovulation is the shedding of the ovum from the ovary via the fimbriae of the fallopian tube. So if the egg fertilize it, better. If the egg does not fertilize it, it will now shed as menstruation at the end of the month. So this is a typical diagram. Are you seeing this slide 23? Are you seeing slide 23?
Yes.
Yes.
Okay. So this is a typical example of one female oocyte. So you can see the follicular cell of the corona radiator. You can see the cytoplasm. You can see the nucleus. You can see the zona pellucida. So, and you can see the sperm there waiting to go and fertilize it. So the sperm doesn't have much job. This job is to fertilize. So that is all about that. So here we say that abnormal meiosis, like most processes, sometimes meiosis makes mistake. So we have what we call non-disjunction occur when two homologous chromosomes move. This is that is under abnormal meiosis. So we say that just as I said, mistake can occur. And then when it occurs, the result is that one daughter cell will be missing, or the chromosome will be 22 instead of 23, and the other will have extra chromosome, which is 24, which is not good. So this type of condition is not is what we call the abnormality. So, and it always occurs. So cells Cells that lack or have too much genetic information will not function properly. And the question is, why will that happen? So the next slide, we also have the non-disjunction. We have trisomy. We have monosomy. So those are abnormal conditions. In trisomy, the condition where there are three homologous chromosomes in the place of a homologous pair. So we see that the zygote with 27 chromosomes, which is abnormal. And in cases of Down syndrome and Klinefelter syndrome, those are examples of trisomy diseases. And we have monosome, the condition where there is a single chromosome in place of the homologous pair. And then we say Turner syndrome, the zygote at this point have 45 chromosomal materials. So this is what happened in the injunction during meiosis. And then you can see that the chromosome is empty. So instead of complete, it gives out to another normal. Then you can see here is a disjunction. So it doesn't share the material equally. So that is in case of disjunction diseases. So another important thing you have to know is how to study this chromosome. A picture of chromosome arranged in homologous pair in descending order by size with sex chromosome placed last. So these are methods of studying cryotyping. Cryotyping, we have it there in the lab, and sometimes I don't know whether they have been sharing or they have done the practical with you. So it is done in genetic lab under clinical chemistry. So where you can do all that. So that is just all. That is the last slide for today. Is there any question? Any question? Me, I'm done. Any question?
I have a question, sir.
Any question? Can you hear me? I hope I'm not muted. Yes.
Yes, I'm not muted. I have a question. On the haploid gamete, uh, we have a formula in the notes. We say like a haploid cell have one N, like one N and two C. So does and also in the note they said like a spermatozoa or a sperm have one N and one C. So how is that possible?
You said they said well like one N.
One N, two C as a haploid cell. Half number of chromosome and two chromatids. Then for the spermatozoa or the sperm, it says like it has one N and one C. So I'm wondering how is that possible?
One N and one C.
Yes.
Right? Yeah. From we in anatomy, we make our things easy. But you see, in genetic, they can have a lot of formula. But if you look at slide, let's go to the slide. Let me see. Are you there?
Yes.
See this slide? Can you see this slide?
We're
Which one?
seeing a slide which is saying karyotyping.
Huh?
We're seeing a slide which is karyotyping.
Yeah, karyotyping. So what you are talking is all about karyotyping, where you have the N, they have N and then two chromatins. Meanwhile, in the male, you can see that because of the Y, you can see the shape of, you can see why they are saying that it's having one chromatin because of what you're saying. So we use this one for us to describe that formula, which is done in chemistry. So this is just the simple reason why it is different. I don't know whether you understand.
Yes.
Yeah, we in clinical anatomy we use structure to explain so many grammar calculations that you can be killing yourself. We try to identify the structure. In genetics, they can do the calculations and bring different formula, but this is what you're trying to say. One N and one for the male, this is the male XY chromosome that have one N haploid, and then a normal with one chromatin material, which is represented by the X and Y. Any other question?
Doctor, I have a question on the disjunction.
Yeah.
What the factor that may influence the disjunction and why does it
You,
happen?
you say?
What are the factors
Right.
that
It's very important. Yeah, there are many factors that can cause disjunction. That is very important question. So many factors can cause disjunction in the sense that um, factors, factors like environmental factor, hereditary factor, nutritional factor, and we also have teratogens from environmental factors that cause this disjunction during critical period of the mother, during critical period of embryonic development. We have critical period where this embryo are developing, which is during the mitotic division. And then the mother at that point, after 2 weeks of pregnancy, she can be exposed to many different things. Some women can be eating too much food. Some they will not even eat the food. Some will smell many things. They want so many things. They have complication, physiological differences. Some like they are exposed to chemicals like the mosquito chemical that kill mosquito at home. Some you see the painting of the room, they paint the room with lead, aluminium, mercury chloride. They mix in so many furnitures. So the women at that point they are exposed to that, and that will lead to those are what we call teratogens. They can interfere during critical period of development to cause what we call disjunction. They can cause trisomy or monosomy. And again, it can also be hereditary problem from generation and generation or environmental exposure, just like what we have in Nagasaki and Hiroshima, where the atomic bomb was the, the Asian, and then that also have caused a lot of disjunction and trisomy and monosomy in terms in the chromosome in the chromatin material. Remember this structure. So everything that I so these these come from molecules. These molecules you sniff them, you breathe them. They are interfere with the proteins or the polypeptide. These polypeptides come together to form what we call the DNA. The DNA come together to form what we call a double helix due to either weak van der Waal, weak hydrogen, or covalent bonds that form to come together to form you with the help of the chromatin material to form these information. These are where interference, these are small molecules where there is interference that will lead to disjunction. We have disjunction, translocation, we have deletion, and many genetic factors that can affect that can result in the abnormal formation, which is the abnormal or teratology that we are going to talk about later. Is that understood?
Yes.
Is that understood? Thank you for that question. Because when you ask a
Lecture.
good question, yes,
I have something to add on the question asked by Arafat. He asked about
okay,
the meaning of one, or the meaning of the 1N and the 2C or 4C during meiosis.
okay,
During meiosis, we start our meiosis with 2N, meaning that we have two chromosomes in our cell. But at the end of the first meiosis, we end with our haploid cell with a single chromosome and 2C. 2C means that even though it is a single chromosome, we have a duplicated DNA component. It means that our component in our haploid cell is duplicated in order to allow the meiosis to take place. That is why we end our meiosis 2 with 1N and 1C haploid cell. Means that does end cell or that gamete will have a single chromosome with an duplicated DNA component in that cell. Thank you.
But it says like haploid has one N2C.
I don't think so.
Check well in the
It
notes.
said that haploid, it's two haploid. You have a haploid from the end of first meiosis and the haploid from the end of second meiosis. The haploid from the end of first meiosis must have one end and 2C, means that its DNA must be duplicated in order to allow the second meiosis to take place. But the
Does
haploid at the end of our gametogenesis must have
but
one end.
does the DNA duplicate
So
during the second meiosis?
no, it deprecates at the S phase of interface.
So why are you saying that in the second meiosis the DNA should duplicate?
No, it is. I have not said it duplicate. I have said duplicated DNA component in order to allow the sharing of those genetic materials between the two haploid cells, which are going to be formed as gamete.
hello, can you hear me now?
Yes, I hear you, sir.
Hello, can you hear me?
Yes, sir.
Somebody was trying
Yes,
to
yes.
someone was trying to add to the 2N and N, the 2N plus 2 to 2C, which is the 2 chromatin, and then the 1N and also 1C, which is the 1 chromatin. Thank you for that contribution, and that is why it's good uh, to read, to interact with the material given so far. You know, I because I try to explain the slides, and as well when you talk about reduction in the we have what we call reduction in cell reduction, the reduction in the number of chromatin, and then this reduction occurred during first and second meiosis, which is different from mitosis, where you have the same number which I've defined earlier. But during meiosis, that's where we have a reduction in this number. Simply that n plus 2c can be mitosis, which is complete number of chromatin materials, and why the 1n and 1c, it could be the stages of meiosis. When you read the material given by Prof, it will add more knowledge on that. I try my best to see how I can summarize in an interactive manner with the hope that you have a good background knowledge on the material. So I thank you so much for that contribution.
Thank you.
So far so good. What I will leave, the message I will leave behind is that gametogenesis or embryology as a whole is a very interesting course, and I always wish you people to be so much interactive with the material. What I'll be doing is to summarize the best and to be more interactive. And then, if you're having curiosity, anywhere you see me in the department, in my office, in the market, you can stop me and ask. I know sometimes some of those things, sometimes some people could be shy. There are issues, but remember that you are going to be the doctor, that you'll be alone, and you're going to serve a community. In fact, you see a lot of problem with family, and then some of this family problem is nothing more than is around gametogenesis. And then because of your primary knowledge, then you can address them. So don't hesitate to stop me anywhere to ask me questions. I think that is the only way, at least one-on-one, face-to-face, or during the practical time. Always stop me, or you can send a chat or WhatsApp. I can explain more better in a practical way, because all I know that we are all mature. So there are nothing I'm saying that is new to you, but I am trying to make it scientifically so that you can use it to solve your problem first, and then the community problem you are going to solve in the future as a doctor to be. Is that understood? Having said that, so I want to close up the class and then prepare it wide. Read wide. I'm going to your TBL is going to come up after cardiovascular and respiratory system development, so that we can have a record for you to compute your CA, your CATs. All right. Bye bye.
Bye bye.